All right, guys, but let's get started. So this is Chapter 9, Patterns of Inheritance. Okay, so you are going to learn about how the genes that you inherit from your parents, what genes you got, whether they were dominant or recessive, how that determines the traits that you have and that we all have and what your children have, okay, or what your children are going to have if you don't yet have children, okay? So it's cool. It's fun stuff. All right, so let's start out with a little bit of a history lesson about who really was the founding father of genetics research, and that is credited to Gregor Mendel. Okay, he began conducting experiments breeding pea plants. Okay, he was a religious man, a holy man. He was a monk, and he lived in an abbey. And so these pea plants grew in the garden of the abbey. And over the years that he was living there, he noticed that there were a tremendous amount of variety in the pea plants. Okay, they could have a variety of flower colors, they could have a variety of seed colors, they could have a variety of seed shapes. Okay, so there's this tremendous variety in these pea plants. And he wondered, why did that happen? How is there this variety? What determines this? Okay, and so to explore those questions, he conducted experiments using these pea plants. Okay, and using these experiments that I'm going to detail shortly, he showed that parents do pass heritable factors, okay, which we now call genes, to offspring. And that determines the traits that are expressed in those offspring. Okay, now pea plants were a really good specimen, a really good species to use for this experiment. Because, first of all, he could control their matings. Do you guys know about flowers? That flowers, most plants can actually reproduce asexually and sexually. Okay? And so, one of the things he did to prevent a flower from reproducing asexually, from fertilizing itself, was he could remove a sex organ from a flower. Okay, like the male sex organ. The sperm-producing sex organ of the flower, he could remove it, and that way he could control the mating. He can make sure that that flower cannot fertilize itself, it can only fertilize another flower. Sexual reproduction. Okay? And then he could look at those different characteristics and quantify the abundance of a certain trait versus another. Okay, so let's take a look at what he did in his experiment. So this is a look at his experiment, okay? So first of all, he was going to breed together, cross, a white-flowered plant with a purple-flowered plant, okay? But first he had to make sure it could not reproduce asexually. So he removes the stamen from the purple flower, and then he collects pollen from the white flower, and then brushes the pollen on the purple flowered plant, fertilizing the purple flower with sperm, pollen, from the white flowered plant. Okay? Then what happened? Fertilization happened, the seeds ripened and matured. Okay? And then eventually he planted all of those seeds from that pea pod, and guess what? All of those babies, all of the offspring, the first generation of offspring, otherwise known as the F1 generation, all had the purple characteristic, no white. Before even moving any further, what does that tell you? What is dominant? Purple. Purple is dominant to white in terms of flower color in pea plants. Okay? And generally that is how it works. Usually dark colors are dominant to light. Okay, and in this case, yes, purple was dominant and occurred in the first generation. All of them were purple. And I'll tell you about that why in a little bit. But first, let's go through an introduction to some terminology. Okay? Okay, the terminology is coming next, but so let's take a look at this. This is what happened, kind of as a little overview of what happened. So one parent is purple, one parent is white, right? The F1 generation was all purple because that was the dominant color. Then what happens is then he took two of those purple flowered plants from the first generation, the F1, and he crossed them together. So he produced offspring in the second generation, the F2 generation, and guess what happened? That white color came out again. Okay? So yes, most of the F2 generation had the purple flower color, but that other color, white, came out in that next generation. Okay? So I'll show you how you would actually figure that out using something called a Punnett square, which is used to solve probabilities of inheritance. Okay, here's this terminology. All right, so first of all, I already talked about this. What are heritable factors? They are genes. And genes exist as alternative forms, okay, called alleles. Most genes are determined by two alleles. You get one from your mom, you get one from your dad when fertilization happens. Okay, all alleles are alternative versions of genes. A genotype. A genotype is simply the listing of alleles for a trait. Okay? So for example, using H's, a genotype could be this. Big H, big H. Okay? A genotype could also look like this. Big H, little h. And a genotype could also look like this. All right? A genotype is simply the listing of alleles for a trait. Okay? This is an allele, and this is an allele. Each of these letters are symbolizing alleles for a certain trait. Okay? This genotype is called homozygous. Homozygous means two copies of the same allele. All right? What about this? Is this also homozygous? Yes, this is also homozygous. Little h, also homozygous. You got two copies of the same allele. However, in genetics, we generally use capital letters to refer to dominant traits. Okay? And we use lowercase letters to refer to recessive traits. Okay? So this is the homozygous dominant genotype. This is homozygous recessive. Okay? What about this one? You got two different alleles here. Big H and little h. That's called heterozygous because you've got two different alleles. Two different alleles, one dominant, one recessive. So that's heterozygous. But what trait do you think is going to be expressed in the heterozygous genotype? The dominant trait. In most cases, there are some exceptions, yes. If you've got a heterozygous genotype, only the trait expressed by the dominant allele will be expressed. Okay? The trait expressed by the recessive allele can only be expressed if you have the homozygous recessive genotype, like little h, little h. Okay? Now, a phenotype. That's another really important vocab term, the phenotype. The phenotype is the expression of the genotype. It is something's appearance. It's what you can see or measure. Okay, a trait that can be seen or measured is the phenotype, and it is determined, of course, by the genotype. Okay? So let's just say that H's refer to your height. Okay? H for height. These letters that we use are pretty random. Okay? They're basically something that kind of helps you to think about a certain trait. So if I want to talk about your height, your stature, I can use H's. But I could also use H's to talk about hair, right? Okay, so just just don't be too concerned about what letters we use for things. You could really use whatever letters you like, okay? But generally we use the ones that sort of make sense in the trait that we're talking about, okay? So if I'm talking about height, okay, and let's just say that tall is dominant to short, okay? Big H is dominant to little h, okay? Big H, allele for tall, little h, the allele for short, okay? So what phenotype is going to be expressed by this genotype, big H, big H? Tall. Okay, so phenotype is tall. What about this genotype, big H, little h? What phenotype? Tall. Okay, remember? Tall is dominant to short. Okay, little h, short. Okay. So does that make sense? I just want you guys to really know the difference between genotypes and phenotypes right now. Okay, genotypes, the listing of alleles or descriptions of the alleles like homozygous dominant, heterozygous dominant, homozygous recessive, okay? And then phenotypes, the expression of the genotype, right? What you can see or measure. You guys clear on that? Okay. All right. Now remember that these genes are on your chromosomes that you inherit from both of your parents, right? Remember this from last week, the homologous chromosomes? Do you guys remember how many total pairs of chromosomes are homologous, completely homologous, which means they have the same size, location of the centromere, and gene loci? How many pairs are completely homologous? Not all 23. 22. That's right. Not the X and Y. They're not homologous. But 22 pairs of chromosomes are homologous. Autosomes. That's correct. 22 pairs of autosomes. Okay? And so take a look. You see an indication on this slide about the location of the genes, the gene loci. Gene loci is just a fancy way of saying locations of genes. All right? So let's take a look. Let's say you got the red chromosome from your dad and you got the blue chromosome from your mom. Okay? Well, on that one gene locus, you got a dominant allele, capital P, big P, from dad, and you also got the same allele from mom. So you are homozygous dominant for that trait, whatever trait that is, coded for by the big P. Okay? Take a look at this locus. Okay? You've got two recessive alleles, one from dad, one from mom. So you're going to have the homozygous recessive trait that's coded for by that gene. Okay? And then take a look at that green locus right there. You got heterozygous, right? You got a big B from your dad, you got a little b from your mom. So you are big B, little b, heterozygous for that trait. Okay? So does that make sense that gene loci reside at the same location on homologous chromosomes, but you could have different alleles on those loci, right? Just like how you've got the big B there and the little b there. All right? Okay, so now let's start getting into the whole meat of this lecture. And we got to start with understanding that the boxes, this right here that you see, this box, is called a Punnett square. And these Punnett squares are probability tables. Okay, probability tables. So unless your outcome is 100% chance that you're going to get a certain outcome, everything is just a probability that something's going to occur. It's not going to tell you a certain event that's definitely going to happen, unless your probability comes out to 100%. Okay? So let's take a look at some of the background behind the setup. How would you set up a probability table, a Punnett square? Okay? We have to start with what happens during meiosis, during the formation of gametes, right? What are gametes? Sex cells like sperm and eggs, okay? Sperm and eggs. We got to figure out what alleles are going to be carried by these gametes, by these sperm and eggs. Okay? So let's take a look. So if we have a heterozygous male, a heterozygous dad, all right, when he makes sperm, 50% of his sperm are going to carry the big B allele, and 50% of his sperm are going to carry the little b allele. Right? Why does that happen? Because of what happens at anaphase, right? The sister chromatids separating, going to opposite poles for each cell that gets created. Okay, that's why. So 50% big B, 50% little b. Female, in this case, she has the same exact genotype for that trait, big B, little b. So when she makes eggs, 50% of her eggs carry the big B, 50% of the eggs carry the little b. Okay, so that's how you set it up. Okay, you can put your male's gametes here, your female's gametes here. All right? Now, the next thing that happens is we have to fill in our probability table. Okay? So what we do is we bring together our possible outcomes of our gametes and we see what's going to happen. And the rule of multiplication applies here. Okay, right? So if we want to consider, what do we get in this box right here? We have to multiply 1/2 times 1/2. And that gives you 1/4, 1/4. The rule of multiplication. Okay? And so you have a 25% chance then of having a homozygous dominant genotype, right? Big B, big B. Okay, you can keep on going. In this box, how do we get this? Well, we got a big B from dad, we got a little b from mom, 1/2 times 1/2 equals 1/4. Heterozygous. One of each, one dominant, one recessive. Okay? And you can keep on going in the other boxes the same way, right? Now, the next step, now that we've filled in our box, is to really analyze our results. And in here, the rule of multiplication, the rule of addition applies, the rule of addition. Okay? So let's see, are there any other squares that are both heterozygous dominant? Are there any other squares other than one that's heterozygous dominant? Heterozygous dominant, big B, big B. No, okay? So what do we get as our end result? Well, we get 25% big B, big B. There's nothing more to be added to that, okay? How about your heterozygous genotype? One of each, big B, little b. 50%, because you have to add 1/4 plus 1/4. This is the same outcome, right? 1/4 plus 1/4 equals 50%, 1/2. Okay? So then you would write up, okay, well, heterozygous genotype, 50%. What about the homozygous recessive genotype, little b, little b? Just 25%, right? 1/4. Okay? So you have to look at the outcome in your probability table, your Punnett square, and figure out your total chances of a certain outcome. What are the chances that these offspring are going to end up with a certain genotype? Okay? And that's how you would do it. You apply the rules of multiplication to filling in the box and the rule of addition for figuring out what you already got in your box and summarizing your result. Okay? All right, and I'm going to show you how this applies to the example I already talked about with the flower color and the purple and white flowers I started with. Do you have a question?
But would this apply to like the actual sex of the child?
If you are considering the sex chromosomes, the same thing. Let me give you an example of that since you're asking, okay? So in terms of sex determination, okay, that's a good question. You guys probably know that what are the odds of having a boy or girl baby? 50%. Well, guess what? Males have what two sex chromosomes? XY. Okay, so males are XY, women are what? XX. Okay, so when men make sperm, 50% of the sperm are going to carry the X, and the other 50% of the sperm are going to carry what? The Y. Female, when she makes eggs, Okay, 50% of the eggs are going to carry the X, and the other 50% of the eggs are going to carry the X as well. Okay, so when we fill in our box, what are we going to get? XX. What about here? XY. XX. XY. So what do we get? What's our rule of addition? Okay, what's the probability of having a girl baby? Okay, 1/4 plus 1/4 equals 1/2, right? 50%. And then XY plus XY, 50%. That's why. That's why the sex ratio is 50-50. And also, girls can do nothing about making boys. Okay, it's all about what sperm got into that egg. Okay? Because 50% of the sperm are going to have the X, 50% are going to have the Y. If the Y sperm gets into the egg, it's a boy. If it's the X sperm that gets into the egg, it's a girl. Men determine the sex of the child. Okay?
What about men who say like I mostly make boys or I mostly make girls?
Every time there was a fertilization event, there was a 50% chance of that outcome.
That statement doesn't apply whatsoever.
What do you mean?
When you have like some families that are mostly male and mostly females.
It still works. It's still the same thing. It just means every time there was a fertilization, there was a 50% chance that it was going to happen. Like, for example, one of my grandmothers, she had 9 total children, right? She had 6 girls in a row, and then she had 3 boys. Okay? But every time there was a conception, a fertilization, there was a 50% chance of either of those sexes.
So but that's what I'm saying. The statement doesn't apply when you hear like men saying, I only make boys.
No, that's just, there's no science behind that. It's not possible. Not possible. Okay. Okay, so now let's get into some principles and laws. So Mendel's law of segregation. This applies to how you set up your Punnett square, your probability table, okay? Mendel's law of segregation states that allele pairs segregate from each other. They separate from each other, okay, during gamete formation. So that each gamete, each sperm or egg, only carries one allele for a trait. Okay? That's all about the behavior of the chromosomes during anaphase. All right? And we can perform a monohybrid cross. What does that word imply? Mono. Mono means what? One. Okay? A monohybrid cross will allow you to predict the probability of inheriting one characteristic, one single trait. Okay? Such as the example that I gave you guys earlier with the flower color, purple and white. All right? So let's go back to that example, and we can do some Punnett squares together to figure out why we got that outcome in the F2 generation with most of the flowers purple, but some of the flowers white. Okay, so remember, we started off with our parental generation. P1 means parental. Parental generation, 1 flowers were purple, and in 1 the flowers were white. All right, so this is how it happened according to how you could set up a Punnett square. So our P1 generation, our parental generation, was true breeding. Okay, if you have a true breeding parents, it means that they are homozygous. Okay, so the purple flowered plant was true breeding, which means it was homozygous dominant, big P, and it crossed with a true breeding white recessive parent. Okay, little p. So now if you have a parent with a homozygous dominant genotype, when that dad makes sperm, all the sperm can only carry one possible allele, right? Big P. It's got no other allele to give. It's only got big P. Okay? And then when you've got the other parent who's homozygous recessive, sometimes I like to use lines underneath undercase letters to help you differentiate them, especially if they look similar, if they're capital and undercase. Okay, so if I draw lines under things, it means it's lowercase. All right. This mom, when she makes eggs, they can only carry what allele? Little p, small p. They're recessively a little p. Okay. So when you set that up, you know, you're going to get all heterozygous offspring, just like you had in your first generation. Remember your first generation? Okay. All purple. That's because they were all heterozygous. And if you want to check and make sure that that's how it's supposed to happen, you can always set up a Punnett square for your F1 generation. All right, so dad, half of the sperm, big P, other half of the sperm, big P. Mom, half of the eggs, little p, the other half of the eggs, little p. And when you bring it together, you get big P, little p across the board. Okay? That's why all the offspring were purple, because they had at least one copy of the dominant allele, big P, that coded for the purple flower color. Okay? Now, where it gets really interesting is when we want to look at the second generation, the F2 generation. Okay, this is the F1. Now we're going to take two individuals from the F1 generation and we're going to cross them together. Okay, that allows us to see some interesting stuff. So let's do over here, we're going to cross a big P, little p with another big P, little p. Okay, so what are we going to put on top of this box? If this is one of the parents' genotypes, what are we going to put here? Big P. What about here? Little p. Okay, here's mom. What are we going to put over here? Big P. What about here? Little p. Okay. Now, how do we fill in the box? What are we going to put in this box? Two big P's. Big P, big P. What are we going to put here? Big P, little p. What about here? Big P, little p. It's always good practice to keep the dominant allele in front of the recessive. Okay? Because it makes more sense like that. What about in here? Little p, little p. Remember what the outcome was? That 3/4 were purple and only 1/4 was white? This is why. Okay? These three genotypes, because they have at least one dominant big P, are going to express the purple flower color, right? And only this one genotype will give you the white color. All right? Now, this is something that you should remember, okay? Take a look at this. Whenever you have two parents who are both heterozygous for one trait, you will always get this outcome. Okay? You get a 3-to-1 phenotypic ratio. That's the ratio of purple to white. Okay? 3/4 are going to be purple, and only 1/4 will be white. 3-to-1 phenotypic ratio will always happen if both parents are heterozygous for one trait. Same thing with the genotypic ratio. If both parents are heterozygous for one trait, you will always get the 1-to-2-to-1 genotypic ratio. Okay, that's just the breakdown of the number of different genotypes you have, right? Okay, so how many out of 4 were homozygous dominant? Just 1, right? 1, big P, big P. Okay, how many were heterozygous, big P, little p? 2 out of 4, right? So there's the 2 in that ratio. And then how many were homozygous recessive? Just 1. Okay, so that's the 1-to-2-to-1 genotypic ratio. Both constants. Yes?
By looking at it, can you determine like which one of the purple flowers are actually the dominant?
Purple flower, the purple color is dominant.
I mean, like, you see how you set it up? where two of them are heterozygous?
Oh,
Can you just look at it and tell which one the
you could.
homozygous dominant?
Homozygous dominant is purple, but no, you'd have to see what would come out in the next generation if you bred them together. No.
And then, second question is a way to scientifically like test out the flower and be able to tell.
Yeah, you have controlled breedings. Like you could cross this genotype with this genotype, and if you get a certain outcome, then you'll know that that was heterozygous and not completely homozygous. Because let's just say if you crossed this with that, all the offspring are going to be what? Purple. But if you cross this with this, you could do a Punnett square to figure out what would happen. We might as well do it since we're on that topic. Okay. So if you wanted to cross a big P, little p heterozygous with a homozygous recessive, because you didn't know that this was homozygous dominant or not, you're going to get a 50-50 outcome. Okay, see this? 50% purple, 50% white.
But you'd only be able to determine it
After you perform the mating.
You can't.
That's correct.
In a laboratory or anything.
That's correct. Well, you could check in a laboratory. You could do genetic testing on what you're working with, but that would cost money. It would be an expense. And it did not exist during Mendel's time. Okay? Okay. All right, there's a lot more to show you guys. All right. Now, let's get a lot more complicated here. Okay? Now, there's another law, the law of independent assortment. Okay? So we know that allele pairs separate from each other, right? We already learned that from the first law. How about Mendel's law of independent assortment? Okay? What this means is that if you've got several traits, or at least two, okay, these allele pairs are going to segregate from each other independently, okay, which means basically that they're not linked. So if you have two different traits, and you've got a dominant allele for that trait and a recessive allele for the other trait, this law assumes that those genes are not linked in any way. Okay, that they're not linked, that they separate from each other and independently, independent assortment. Okay, this is going to make more sense when I show you how to actually perform this independent assortment to figure out what alleles the gametes are going to carry. Okay, so we do this, we apply Mendel's law of independent assortment when we are tracking the inheritance of two traits at the same time. Okay, so you know that simple 4-box Punnett square I just showed you how to work problems with? We need a big 16-square Punnett square box to do this. Okay, this is the hardest type of problem that you guys will have to solve. Okay, so that's why I'm giving it to you now and not at the end of the lecture. All right, so let's talk about this. It's called a dihybrid cross, because di means what? Two. Two traits at the same time. So we need a dihybrid cross. Now in this case, we're still dealing with flowers because they're easy. Pea plants and flowers. Okay. So we are starting with a true breeding parental generation. Okay. One parent has two dominant traits, round seeds that are yellow. Okay, round yellow seeds. For round, you need to have at least one big R. Okay, and then for yellow seeds, you have to have at least one big Y. That's the dominant trait, round yellow seeds. In this case, the parent is homozygous dominant for both of those traits. Okay, that parent is breeding with a parent who is completely homozygous recessive for two traits. Okay, little r will give you wrinkled seeds, and little y will give you green seeds. Okay, but what's going to happen? Just like what happened when we crossed our purple and white flowers together, okay, what are you going to get? You're going to get offspring in the F1 who only look like one of those parents, right? Remember that? All heterozygous. In the F1 generation, only plants with round yellow seeds have been produced because you have at least one of each dominant allele. That would be the genotype that results from a cross between those two parents. Big R, big Y, little y. Okay. You want to know why? Okay. Well, let's just do a really small little Punnett square box before we get into the big one. I want to explain how this has happened, okay? In a gamete, in a sperm or egg, you are only going to have one copy of an allele for each trait, okay? So if you're dealing with big R, big Y, okay, you want to figure out what are the possible allele combinations in the gamete, okay? And to do this, we usually do something that we use in math called foiling. It's sort of similar to that. So we do this. We go, okay, this is going to connect to that. That's going to go to this. Then we go here. See where I'm going? A parent who has this genotype, homozygous dominant for two traits, will only be able to make gametes that have this combination, big R, big Y. Everything else is the same, right? Only one possible gamete combination. Same thing for the other parent who's little r, little y, right? If you do that, you're going to always get the same result. Okay? So if you wanted to see what would happen if you put these two gametes together, you could do a really kind of short little Punnett square box and say, okay, big R, big Y, little r, little y, and you can go big R, big Y, little y. Okay? That's unusual to have such a short little Punnett square box. That's only because one parent's completely homozygous dominant, the other parent's completely homozygous recessive. Okay? The fun begins with the 16 square Punnett square box when we want to now cross two of the offspring from the F1 generation together who both have this genotype. Okay? So, if you have this genotype, big R, big Y, little y, you got to now figure out what allele combinations would end up in the gametes. All right? So, let me go ahead and erase this, or at least I'll move it over here. How about that? Okay? So we are going to now conduct a big dihybrid cross between a big R, big Y, little y, with another big R, big Y, little y. So to do that, we're going to set up a big 16 square Punnett square box. So you draw a big box, put 3 lines down the middle and 3 lines horizontal. Okay. Now we're going to go ahead and foil again so we can set up our Punnett square. We got to figure out what alleles are going to be carried by the gametes. All right. So let's start off with this parent. All right. So we're going to go big R to big Y. And we'll put that one right here. Then we can go big R to little y. Then we'll go little r to big Y. And then little r to little y. Did you see that? Okay. So I've set up the gametes. Let's just say that that's the dad. I set up the possible combinations of alleles in all the different types of sperm that dad can make. He can make 4 different types of sperm right there. Okay. Now we got to do the same thing for mom. But conveniently, she has the same exact genotype, okay? So, but we can foil again to make sure that we get the same consistent results. So we're going to say big R to big Y, big R to little y, little r to big Y, and little r to little y. All right. So now help me fill it in. Guys, let's hear what you say should go in here. Okay? So what should go in here? What? Yeah, big R, big Y, Okay? I'm going to keep on going, but think about what should be in these boxes because I got to get this done. It's going to take me a little bit. All right? So here we go. Big R, big Y, little y, right? Big R, little r, big Y, big Y, big R, big Y, little y. You got to be careful with your penmanship to make sure that you don't get a big and a little confused, okay? Or write it improperly. Here we got big R, big Y, little y. Are you guys with me? Big R, little y, little y. Big R, big Y, little y. Big R, little y, little y. Big R, big Y, big Y. Big R, big Y, little y. Little r, big Y, big Y. Little r, big Y, little y. And big R, big Y, little y. Big R, little Y, little y. Little r, big Y, little y. And last but not least, little r, little y, little y. Whew, that's a lot of work. Okay. Now we have to interpret our results, okay? So let's figure that out. Now, I started out with two completely heterozygous parents, right? Both parents were heterozygous for two traits, okay? So just like how if you start with that with a monohybrid cross, you have a consistent phenotypic ratio, you are also going to have a consistent phenotypic ratio in this case as well. And in this case, it is 9 to 3 to 3 to 1, just like it says in the slide. Okay? Now let's figure out what that means, and let's identify the genotypes that correspond to that ratio. Okay? So basically, though, just to let you know, that means that 9 out of, how many possible outcomes do we have here? 16. 9 out of 16 are going to have both.
Homozygous dominant.
Well, not necessarily homozygous. They're going to have both dominant traits. But they could be homozygous or heterozygous. Okay, so in fact, let's stop there and let's find them. Okay, that's a good segue. So let's look for the 9 out of 16 that are going to have both dominant traits. Okay, and to have that, they have to have at least one copy of big R and at least one copy of big Y. The second letter does not matter. Okay? So we're going to have both dominant traits, which was what? Remember? Round yellow seeds. All right. So let's find them. So does this one work? Yes. What about this one? No way, right? What about this one? No. Yes. Yes. Do we have 9? Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 out of 16 are expected to have both dominant traits, round yellow seeds. Okay? Now, we're going to have 3 out of 16 that we expect to have one dominant trait and one recessive. Okay? So let's say that they've got the dominant round trait, but they're going to have wrinkled, they're going to have green seeds. Okay? So round seeds that are green. So one dominant trait, one recessive. So let's find our 3 that fit this bill. Okay, what about this one? Yes. How about this one? This one? There we go. 3 out of 16. Dominant for the R gene, giving you round seeds, but recessive for the Y gene, giving you green seeds. Okay? Let's keep on going. We got to find 3 more. In this case, it's going to be the opposite, where you have the recessive shape, wrinkled, but you're going to have the dominant color, yellow. Okay? So wrinkled yellow seeds. All right, so where are they? There's not much left. So what's this? Yes, that works. What about this one? Yes. How about this one? Yes. Okay, so 3 out of 16 have the phenotype wrinkled yellow seeds. And then we have our one lonely one left. 1 out of 16 is completely homozygous recessive for both traits, giving you wrinkled green seeds. Okay? How do you like that? Was that fun? Oh, stop it. You'll get practice. Okay. The 9 to 3 to 3 to 1 phenotypic ratio. But see, if you get asked on a test, what is the phenotypic ratio of 2 parents who have, who are both big A, little b, big B, little b? Okay? Big A, little a, big B, little b. What would you say? 9 to 3 to 3 to 1. If they're both heterozygous, you don't have to do all that work. You already know what the answer is going to be. Okay? Think about that. All right. Here is another example that you could work just to give you guys some extra practice for over the weekend before we meet again on Tuesday to do more practice together. But you could always do a Punnett square with these different letters, big B, big N, little n. Okay, 2 parents with that genotype, and you should get the 9 to 3 to 3 to 1 phenotypic ratio when dealing with the inheritance of coat color and blindness in dogs. Okay, so try it out. All right, in this case, black is dominant to chocolate, and the blindness trait is recessive. Okay, so only dogs that are little n, 2 copies of the recessive gene are going to be blind. If you have 1 big N, you're normal. Okay, so I'm not going to do that again because it would just take too much time. Okay, that's more practice for you guys to do at home. So do a cross with big B, little n, times a big B, little n, big N, little n, and you should get the 9 to 3 to 3 to 1 ratio when you work it out. Okay. Okay, now let me talk about some exceptions here. Linked genes do exist. And this was something that Mendel really did not know a whole lot about. Okay? Linked genes do actually exist. So it does sort of counters his law of independent assortment. In some cases, genes do not separate independently of each other. Okay? In some cases, they are linked. And this experiment that was performed by Bateson and Punnett, who the Punnett square was named, who was basically the reason for the naming of the Punnett square, found an example of the inheritance of plants and plant traits in which actually there was gene linkage, and that defied Mendel's law of independent assortment and defied the predicted 9 to 3 to 3 to 1 phenotypic ratio. Okay, so in this case they had parents where one plant had both dominant traits. Okay, purple flowers and long pollen crossed 2 plants that had the recessive traits of red flowers, round pollen. And sort of ironically, in the second generation, you didn't have the 9331. Instead, the majority of the F2 generation did have both dominant traits, but the second most abundant characteristic, the second most common characteristic, was both recessive traits. Okay? Red flowers, round pollen. So the dominant alleles in this case were linked together, and the recessive alleles were linked together. All right? So that defied Mendel's law of independent assortment. You will not have to solve problems with gene linkage, though. Okay? But this was the outcome of their experiment. So if Mendel's law of independent assortment held up to be true, this is what we should have gotten. according to the 9 to 3 to 3 to 1 ratio. Okay? But we didn't. This was what was observed from the cross between those two parents. Okay? That yes, the majority of the offspring did have both dominant traits, but the second most abundant characteristic was both recessive traits. Right? Whereas, according to 9331, both recessive traits should be the most rare. Okay? That's just a little exception. And just as an extra sort of way to go after this thing I'm talking about, linked alleles, though, the huge benefit of meiosis, which leads to genetic variety and diversity, is because of crossing over. Crossing over can separate linked alleles. Okay? And let me ask you this. When does crossing over happen? During what phase of meiosis? What? Prophase what? 1. Prophase 1. Due to crossing over, which happens at prophase 1, that will actually separate linked alleles. Crossing over can separate linked alleles and can lead to genetic variation and diversity. Okay. Now, before I start getting into more problems that you guys can figure out how to solve, just one more thing is what a genetic map looks like. This is a genetic map. It shows you the location of a certain trait on a chromosome and how close in proximity they are to other traits, other gene loci. Okay? And basically, what is depicted here are recombination frequencies, which really shows you what is the chance that these alleles will be separated from each other in crossing over to produce a recombinant. Okay? Let me just go back to the previous slide. This is a recombinant right here. This is the result of crossing over. This is the result of crossing over. These are recombinants. Okay? So this next slide is wanting to ask, what is the frequency of recombination happening? Okay? Basically, if they are farther apart on the chromosome, they have less of a chance of being inherited together, and the greater 17% recombination frequency. Okay, they have a higher chance of being separated by crossing over. Okay? If they are closer together on the chromosome, okay, they tend to have a lower recombination frequency. Okay, a lower chance of being separated by crossing over. Okay, so genes that are located close together on chromosomes tend to be inherited together. The further apart they are, the more likely they are to be separated by crossing over. Okay? All right, now let's get into some more variations on Mendel's laws. There is this case of incomplete dominance. Incomplete dominance means that neither allele is dominant over the other. And instead, when you have a heterozygous genotype, you have a mixture, a blending of the two parental genotypes. Okay, so in this case, we've got one true breeding red flower, big R, crossing with a true breeding white flower, WW. Okay? And when you bring that big R and that big W together, because of incomplete dominance, neither allele being dominant over the other, you get a new phenotype. That's a blending of those two genes. So what happens if you have paint, red paint and white paint, and you mix it together? What do you get? Pink paint. Okay? So then heterozygous flowers, RW, are going to be pink. A blending. Okay? This does not happen incredibly often, but there is this case where you can have this mixture, this incomplete dominance in certain traits. Okay? Now, of course, what happens if that whole first generation is pink, RW, and then you cross the two RW pink flowers together? That's what we've done here. So guess what? Pink flowered male, pink flowered female, and when you fill out the Punnett square, guess what? The two original parental phenotypes are expressed. 25% red, big R, 25% white, big W, and then 50% though are going to still express that mixed blended phenotype because they are heterozygous, RW. Okay? So if I ask you a question that says, if red flowers cross with white flowers and all the offspring are pink, this is incomplete dominance. That's what you should tell me. Incomplete dominance, as opposed to complete dominance. These other things that I've been talking about are complete dominance, right? If you've got a big R and a little r, only the trait in big R is expressed, that's complete dominance. Okay? This is incomplete dominance, which results in a new phenotype that's a blending of the original parents' phenotypes. Okay. Now, I do want to spend a good amount of time on talking about blood types, because blood types are really cool, okay? But they can also be a little bit confusing sometimes, okay? So consider this fact, that there are more than 2 alleles in the human population for blood type, right? There's the A allele, there's the B allele, and there is the O allele, 3 alleles. So this is a case of multiple alleles. But each person is only going to carry 2 of these alleles. All right? However, because of the dominance and recessive relationships going on here, we do have 4 possible blood types, right? You can have A blood, B blood, AB blood, or O blood. And I'm going to show you why. But the whole reason why the AB blood type exists is because the A and B alleles are co-dominant to each other, which means that both alleles are expressed. Okay, this is not a new phenotype technically. It means that both alleles are expressed. Okay, and there's going to be a really useful picture on the next slide that illustrates this. All right, so let's talk a lot more about blood types. Okay, this is a very helpful diagram. So the phenotypes, when you have your blood type tested, which do you guys know that one of the best advantages of going and giving blood is that you get to find out your blood type. Not only are you doing a good thing, right, to help out the blood banks, but you also get to find out your blood type for free. Because on its own, if you wanted to find out what blood type you are, it would cost a lot of money. Okay? But if you go and donate blood, you usually find out for free. Okay? And it's a useful thing to know. All right. So, but when you get your blood type tested, what they tell you your blood type is your phenotype. Okay? So phenotypes for blood type can be O blood, they can be A blood, they can be B blood, they can be AB blood. Okay? Now, in terms of genotypes, okay, we're going to list out the possible genotypes that can give you these phenotypes, all right? But when considering this, you should remember that the alleles A and B are dominant. They are dominant, and O is recessive. Okay, the O allele is recessive, A's and B's are dominant. Okay? So if you are O blood, what does your genotype have to be? O. You got to have 2 copies of O. Okay? It can be symbolized using those little undercase I's, but I think it makes it easier to do your Punnett squares just using the O's alone. Okay? It makes it less confusing. So anytime you see an undercase I, that means O. Okay? It means a lack of carbohydrate attached to your blood cells. Okay? See how in the picture there's no carbohydrate on the O blood cell? That's because O blood does not have carbohydrates attached to their blood cells. Yes?
To find this out for the blood cells, would you use the 4 squares or the 16 squares?
Just 4. Just 4. Okay? Make it simple. We're not going to do anything else in 16 squares. Okay? Everything else will be simple monohybrid crosses. Okay? But there's special rules for blood types, okay? Now, if you are type A blood, you probably do not know if you are homozygous for A blood, AA, or if you are heterozygous for A blood, AO. Okay? So if you're A blood, you could have two possible genotypes. You could be homozygous for A or heterozygous for A. Okay? Remember that O is recessive. So if you're AO, you're A, because A is dominant to O. Okay? If you are B blood, it's the same situation. You can be homozygous for B blood, or you could be heterozygous for B blood. You probably don't know. Okay? If you're AB blood, you're AB, right? Your genotype's got to be AB. The only reason why this blood type exists is because the A and B alleles are equally dominant. They're co-dominant to each other. All right? So take a look at that. This is really important to remember, okay? Because if I ask you a question, well, let's do an example right here, okay? Let's say we're going to cross a man who is heterozygous for A blood, okay? So we got a dad who's heterozygous for A blood. What's his genotype? AO, okay? So half of his sperm carry A, half the sperm carry O. We're going to cross him with a woman who is heterozygous for B blood. So what's her genotype? BO. So half of her eggs are B, half of her eggs O. So let's see what we got here. What are we going to get? AB, BO, AO, and O. Take a look at that. That's awesome. A cross between a parent who is heterozygous for A and heterozygous for B will give you a 25% chance of having babies with any of the blood types. Okay? Before I mentioned this example, I had a student once who really freaked out about that. Okay? She was like, well, but I'm O blood, and my dad is A, and my mom is B, and my sister is AB, so is one of my parents not my parent? Kind of thing. Is my paternity in question? And I was like, I had to think about it for a second. Of course, I don't know for sure, but I was like, this is how it's possible. And I said, well, probably your mom is heterozygous for A, your dad's heterozygous for B, okay? There you are. There's your sister. It's possible. I don't know for sure, but yeah, it's possible. 25% chance of any of those blood types popping up in that generation. Okay? O blood. Do you guys know what happens if you get the wrong type of blood? Okay? Your blood will clot, and you can have serious health implications or die. Okay? So take a look, though. One of these blood types is a universal receiver, and one of them is a universal donor. Okay? And you can tell by what it is based on that diagram right there. It shows you what condition blood would clot. Okay? So who is the universal donor? O blood. Okay? For example, I have type O blood. O blood is the most common blood type amongst human beings. Okay? And O blood is the universal donor. Anybody can receive O blood because you don't have those carbohydrates on your cells, so you don't have the antibodies to fight them. Okay? And so anybody can receive O blood, not a problem. And AB blood is going to be your universal receiver. They can get any type of blood. Okay? Any type of blood people with AB blood can get. And no clotting happens. But really bad situations happen if you're O blood and you get A or B or AB blood, forget it. Okay? Same thing with those couple other examples. Okay. How much time do I have? All right, I'm going to finish up a couple more examples here, and then we'll finish it up the rest on Tuesday. So inherited human disorders. So in some cases, genetic diseases that people have can be because of dominant inheritance, where you only need to inherit one copy of the dominant allele to have a trait. Okay, it could be a harmful trait. And so like the allele that causes dwarfism, okay, so reduced stature, okay, tend to have some abnormalities in spine and skeletal structure. It can lead to a lot of pain. that is a case of dominant inheritance. So let's take a look at, I'm going to go back to this slide, but let's look at this example. Okay? So if you have two parents that have dwarfism, but they're heterozygous, big D, little d, big D, little d, because they have the dominant trait, they have the condition, right? But if they have babies, and they're both heterozygous, they do have a 25% chance of having a normal, not affected by dwarfism child. Okay? But 3/4 of them are predicted, do have the probability of having dwarfism like their parents. Because all you need is one big D allele to have it. In the case of recessive inheritance, you need 2 recessive alleles to show the disease, to show the trait. Okay? And so if both parents are heterozygous and they carry the allele, they can pass that trait on to the parents, to the offspring. They can pass that trait on to the offspring. Okay? Generally, the probability of recessive alleles does increase with inbreeding and mating between close relatives. Okay, so here's an example of recessive inheritance. Okay, so you can see that if we're talking about deafness here, okay, you've got 2 parents who are normal, they can hear, but they both carry the deafness allele. When they have babies, they have a 25% chance of having a child who's deaf. All right. Okay. Here is a look at just some examples of genetic abnormalities. This kind of relates to that assignment that you guys have to do, your other writing assignment. So you can research a different genetic disease, or else you can even pick one of these, okay, and talk about it. But I'm going to kind of leave that discussion for that assignment. All right, so these are just some examples of conditions, whether they're recessive or dominant disorders, what they cause, and then the proportion, the frequency of these occurring in populations. Some of them are more common in different ethnic groups and racial groups than others. Okay. Okay. Pleiotropy. Pleiotropy means that you have one gene that influences many characteristics. So the gene that causes sickle cell is an example of this, pleiotropy. Okay? One gene that influences many characteristics. So the gene for sickle cell disease affects the type of hemoglobin you're going to produce. It's going to affect the shape of the red blood cells. Okay? It specifically causes some red blood cells to sickle. And it can cause anemia and organ damage, especially if untreated for a long period of time. But it is also related to the susceptibility to malaria. This is pretty cool, okay? Now, where in the world is malaria, a disease transmitted by mosquitoes, most common? Africa. And any tropical areas of the world. Okay, but in Africa, okay? And so what group of humans is there a higher incidence of the sickle cell gene? Amongst people of African descent. Because for people who live in Africa, at least carrying this gene has benefits. Because if you carry this gene, you have resistance to malaria. And malaria is a scourge that wipes out a huge amount of people around the world every year. Okay, it has such a huge negative impact on the human population. Okay, and this gene actually exists due to an adaptation to survive malaria. Okay, so let's take a look at that. Okay, so let's take a look at the breakdown of genes. Sickle cell is from recessive inheritance. So to have sickle cell disease, you have to be homozygous recessive, little s, okay? Sickle cell. But the other genotypes that are possible, right, are big S, and also what? Big S, little s, okay? Now, if you are big S, you don't have sickle cell disease, you're normal, But if you get malaria, you're probably going to die of malaria. Okay? Now, the person who is little s is going to be negatively affected by sickle cell disease. Okay? However, they are going to be almost immune to malaria. Okay? The best thing to be is heterozygous, because with this, you are still considered normal in terms of your cells, not sickling, and you are resistant to malaria. So over generations, amongst people who live in malaria-plagued regions of the world, this genotype has become the most persistent and abundant, because these are the people who tend to have the most babies and survive longer and pass this gene on to the next generation. It's called a heterozygote advantage. You're a carrier of the recessive allele. So you have that resistance to malaria, but you do not have the negative impacts of sickle cell disease. Yes?
So the SS, the third one, are those both recessive?
These are both recessive. Little s, with thus the line underneath. Okay? All right, guys, I guess I will end it right there. I only have really one major topic left to cover, which is sex linkage. Okay? So thank you for your attention, and I will see you guys on Tuesday. Where I left off last Thursday was on the topic of pleiotropy. Remember that? The fact that one gene can influence many traits. And so one of the examples was the gene that causes sickle cell disease, but that's also related to a person's susceptibility to malaria. Okay? One thing that I wanted to mention before I wrap that up is that the incidence of heterozygosity of the sickle cell gene in the African American population used to be 1 in 10. Okay? 1 in 10. What does that mean, heterozygosity? If someone is heterozygous for the sickle cell gene, what is their genotype? And we use S's for that. What is the genotype of a heterozygous condition for sickle cell? Well, using S's? Big S, little s. Okay? So in African Americans in the United States, the incidence of heterozygosity for the sickle cell gene used to be 1 in 10. Okay? That's where it started. But then over time, since malaria is not being a selective force in the population here in North America, the ratio is actually dropping. It's now 1 in 12. Okay? So the odds of having people who are carriers of the sickle cell disease is actually declining. And so now it's not 1 in 10, it's 1 in 12. Okay? Because malaria is no longer a selective force that's keeping this allele in the population. Okay? So it's interesting. Anybody have any questions on that? Okay. Another thing that I just want to mention very briefly is the concept of polygenic inheritance. Okay? In most cases, I've discussed, one gene to control one trait, right? Or in the case of maybe a dihybrid crosses where we're talking about 2 traits and tracking the inheritance of those 2 traits right at once. The concept of polygenic inheritance is a bit more complicated. This is a situation where many genes influence one trait. It could be 3 separate genes all together influencing one trait, like eye color or skin color. And that's, in fact, how it is. So an example of the genotype for eye color or skin color would look something like this. Okay? So you've got 3 separate genes influencing that one trait, the A gene, the B gene, and the C gene. All right? Now, to predict probabilities of inheritance for eye color or skin color, being that we have these complex genotypes, guess what kind of a Punnett square you'd have to make to determine probabilities of inheritance between two individuals with this genotype? Okay, so the dihybrid one is a 16 square Punnett square box. You know how many boxes you would need for this? 64. Am I going to ask you to solve problems using a 64 square Punnett square box? No. Okay. Don't worry about it. However, if you do want to practice it, there is a really cool link that I have to your D2L page. It's called Genetics Practice Problems. Okay? And there's all sorts of practice problems that range from simple monohybrid crosses to dihybrid crosses, okay, to ones like this where you can calculate the probability of inheritance with skin color using a 64 square Punnett square box. Okay? Trihybrid crosses. So you might want to check that out if you really want to practice your skills. Okay, but that's why there's such a range in skin color tones and eye color, right? Think about all the eye colors, right? There's black, there's brown, there's hazel, there's green, there's blue, and there's different intermediates between that. Okay, that's why, because it's a case of polygenic inheritance. Okay, now let's look at sex linkage. This is the last type of a problem that you will have to be able to solve, is sex linkage problems. And those of you who are in lab class already know from yesterday a little bit about this, okay? But those of you who are in lab tomorrow or later on today, you'll get some practice with that. But as an introduction behind that concept, we have to review the sex chromosomes, right? So the sex chromosomes are X and Y. They are not homologous. They're not the same size, right? You can see that the X chromosome is huge compared to the Y chromosome. All right? And males are XY. They get one of each chromosome. They get the X chromosome from their mom, and they get the Y chromosome from their dad, and that's what made them a male. Okay? Females, in contrast, are XX. One X from mom, one X from dad. Now, some genes are linked to specific chromosomes. Those genes that are either linked to an X chromosome or to a Y chromosome are called sex-linked genes. And so X-linked genes, genes that are linked to the X chromosome, and there are many, okay? And that's because the X chromosome is huge. And there is so many additional traits and characteristics that are linked to that X chromosome. Okay? In contrast, the Y chromosome, you'll notice, is very, very small. There's not much extra stuff linked to the Y chromosome. Mostly the Y chromosome codes for maleness. Okay? But X-linked genes can be passed from mother to son, okay, or mother to daughter. X-linked genes can also be passed from father to daughter. And Y-linked genes, though, have to be passed from father to son. They cannot pass to daughters. Okay? So let's take a look at some examples of sex-linked genes and their ramifications. Okay. A lot of disorders, recessive sex-linked disorders, tend to be linked to the X chromosome. And the reason is, like I mentioned before, the X chromosome is huge compared to the Y. Okay? I don't know if you guys can see this poster that well over here, but it has all the different chromosomes that we have in the human genome that we get from each sperm and egg when fertilization occurs. And it has a listing of all the different traits that are coded by each chromosome. Can you see this? This is the whole list for the X chromosome. This is the Y. Okay? So you can see that there's a whole bunch of stuff coded for in the X chromosome, and very, very little other than maleness in the Y. Okay? So there's a lot of extra stuff linked to these X chromosomes, and some of them are harmful conditions. Okay? One of them is hemophilia, a recessive X-linked condition. What does hemophilia cause? You guys don't have any idea? Somebody has to know it there. Don't be shy. What? Something about blood, right? Yeah. The inability for blood to clot is the problem. And so basically people who are hemophiliacs, if they get cut, a serious cut, they can actually bleed to death from that because of the lack of blood clotting. Okay, you know how if you get a cut, you put pressure on it and it stops? People who who have hemophilia, that doesn't happen. Okay, they need some extra special medical attention. So hemophilia is an X-linked disorder, recessive condition. Color blindness also, and Duchenne muscular dystrophy. Now all three of these examples of X-linked conditions are more common in males than females. And this is the reason, because males only inherit one X chromosome, right? From their mom. And if the recessive allele is linked to that X chromosome, they've got the condition. Females can have these conditions too, but they'd have to inherit two X-linked chromosomes, two X chromosomes that both have that recessive allele linked to the X chromosome. Okay? So for example, if we're talking about hemophilia, this would be the genotype of a male with hemophilia. Okay? Undercase allele, recessive, remember that? This male would have hemophilia. In order for a female to have hemophilia, she'd have to have inherited this. The X chromosome with the sex-linked recessive allele for hemophilia from both mom and dad. And that's very unlikely. It's very uncommon, which is why these conditions are much more common in males than females, because males only need to inherit one affected X chromosome to have the trait. Okay? Now, let's look at this pedigree here. This is a pedigree, this sequence of circles and squares that are all kind of connected. It's like a family tree, all right? And pedigrees are used to trace the inheritance of a trait across generations. This is especially useful when you have genetic conditions that you want to be able to track in your family. So if you have a half-filled circle or square, that means that somebody is a carrier of the trait. And if they're a carrier, what's their genotype? Are they homozygous or heterozygous? Heterozygous. Carriers are heterozygous. Okay? And if it's a white circle or square, that just means they don't have it at all. They're not carriers and they're not affected. And if you have a completely closed circle or square, that is a person in the family who has that recessive X-linked condition. Okay? So let's take a look at this pedigree right here. Okay, who do we start with? Who is Queen Victoria and Albert? Where were they the royal family from? History, guys, come on. What do you think? Guess, royal families, what country still has royal families and stuff? England, right? Yeah. They were the Queen Victoria was the Queen of England, and her husband was Albert, her first cousin. Okay? Yeah. Now, they had lots of kids, a whole boatload of kids. Okay? We're going to follow the pedigree, though, to their daughter Alice, who just like her mother, Queen Victoria, was also a carrier of a recessive X-linked condition, hemophilia. Therefore, what was her genotype, do you think? What do you think both Queen Victoria and Alice's genotype? Okay? Okay, what kind of X? What kind of H? So X what? X what? She's a carrier, so heterozygous, right? So X, what kind of H? Not small. Big H, and then this X is what? Little h. Okay? This is the genotype for this condition of both Queen Victoria and Alice. Okay? They are carriers, so they are heterozygous for the condition. They don't have hemophilia themselves, but they carry it. Now, Alice and Louis, they had a daughter, Alexandra, who just like her mother and grandmother also was a carrier of hemophilia. Now, she married the Tsar of Russia, Nicholas II. And this is them and their family right here. So this is Alexandra, that's Tsar Nicholas of Russia. And in fact, this is the last royal family of Russia. Okay? Anyone know what the name of this family is? Like what their last name is? That's probably a tougher question. This is where the whole legend of like Rasputin and all that comes in. And Anastasia, okay? Anastasia, in fact, is right here, okay? So, anybody know yet? The Romanovs, okay? The Romanov family. This is the Romanov family, the last royal family of Russia. And in fact, they were all murdered right after this picture was taken, okay? After the communist takeover of Russia and it becoming the Soviet Union for that period of time that happened. Okay, so this is a really famous picture, and it's a really famous case. Okay, the whole thing with Rasputin came about because Queen Alexandra had hired Rasputin. He was kind of like a magician, sort of a mystic man from the Ural Mountains. And it was believed that he basically told the royal family that how he can cure their son, Alexei, who has this horrible condition where he just bleeds and it doesn't stop, okay? Because after all, Alexei, he was the only male of the family and he was expected to inherit the crown and be the king, okay? But he was very weakly because he had this condition, this genetic condition. Of course, there was nothing that Rasputin could do to cure him of this condition because it was in his genes, it was genetic. Okay, but that's sort of a whole story unto itself, is the influence of Rasputin on this family. But as a whole, okay, we can see that Alexandra then passed on the allele for hemophilia on her X chromosome to Alexei. Okay, and that's why he has that condition of hemophilia. And so Alexei's genotype, therefore, is what? X little h, y, okay? Now let's see why this happened, okay? So we're going to do a little Punnett square, and we're going to cross Alexandra's genotype, X big H, X little h, with Tsar Nicholas, who was normal. He was not a carrier of hemophilia. And so if he's not a carrier of hemophilia, what's his genotype? What kind of H on his X? Big H, Y. Okay, so this is the cross we're going to do. So we just have to do a simple monohybrid cross, a 4-square Punnett square box. Okay, let's start off with mom's genotype. Okay, so when she made eggs, half of her eggs were what? X, big H, and the other half of her eggs were X, little h. Okay, Tsar Nicholas, when he made sperm, half of his sperm were X, big H, and the other half of his sperm were Y. Okay, so when they had kids, they had a 25% chance of having a normal, non-carrier daughter, and they had lots of daughters, okay, 4 of them. They had a 25% chance of having a normal but carrier daughter, okay? They had a 25% chance of having a normal son, and they had a 25% chance of having a son with hemophilia. And this is Alexei right here. Okay? So that's how you would do a sex linkage problem. You would set up the parental genotypes, and you would set up your Punnett square and fill in the box to predict the probability of inheriting a sex-linked condition. Okay? All right. Well, that is the conclusion for the Chapter 9 lecture.